I'm going to talk about some changes that we're in the process of making to our export options to export the results that your queries produce. So currently, Randy touched on it earlier, that we have this option over here, and it pops up this little window where you can export to CSV files only in one of two formats, either all the fields that we determined to export and then all the fields with IRIs. This is a little bit limiting from the standpoint of it only produces CSV files, and it only produces the CSV files with these particular fields that it's kind of an all or nothing shot. So I don't know if you're familiar with CSVs. They come across as comma delimited, so they're this way, and they're readable by programs and by something like Excel, but they're not very human readable or really useful outside of those formats. So here it is in Excel. Oops, this one. So you see that it pops up, and we've got these two header formats, one with a section like epitope or related object you can see here, and then a subheader that describes that particular field's data. But as you can see, because it's only in CSV format, we are limited to what we can display here and how Excel can display it to the user. So that came up with the fact that we are in the process of redesigning it and producing this format or this export feature here, where you can choose your file format. Excel, this is a native Excel file that it produces by itself. The CSV, which is the current format, a TSV, which is a tab separated format, which is the same as CSV, just tabs instead of commas. And then a JSON format, which is a more programmatic structure that is used on the web to transport data efficiently. That would be something you could use to import into tools that you use or something else uses. Additionally, what we're given the option that we're having the option is to change the headers from that double format that I showed a little bit ago to a single format as well, which combines the section and the header name into one field, which is more standard for importing into various programs or file structures. And then also the ability to have no headers if you just want the straight data to import more easily if that's what's required. So you don't have to go in and remove any data from the export itself. And then here's the meat and potatoes of everything. We're allowing the ability to fully customize what fields your export will contain. So this is a kind of combination between this section down here that I'm showing and then this export type drop down. So along these columns to include, you can pick and choose whatever pieces of data from the website you're interested in. So say if you're not interested in the starting and ending position, you can get rid of those, and then they won't be in the export and they won't be taking up space, and you won't have to deal with them in whatever you're writing or working with. In addition to that, you can see that you have all the different fields that we have available for the exports in this option, a nice convenient list. You're going to be able, you can completely remove sections if you'd like. You can completely add them. You can pick and choose whatever you want. But as well with that, we have these export types, which are going to be predetermined exports that the IEDB team kind of sets up of sets of data that could be commonly used by users. So for example, what we have the full ones, which is everything. The default, which kind of matches up to what we currently have as our exports, which doesn't, you can see, for example, that doesn't include the IRIs because our current one the default one does not have the IRIs, but then you also have the default with IRIs, which, as you can see, includes the IRIs here. And then we're also looking into adding other export types here for various sets of fields that we determine is commonly used. It's going to be dependent on user feedback and usage. We're also working on having the ability to directly export the fields as you see them on the screen here, which we do not have right now. We don't have the ability to easily pull this exact data off the screen, and we're looking into adding that to the ability to export it. So one of the big things with this is it also allows us to natively create Excel files as opposed to the CSVs, which you can see here is the same export, just a smaller data set because it's development. As my previous one, you can see that it's formatted a little bit nicer. It gives us some more flexibility to show the data better, and it can be manipulated in Excel a lot easier because it is a native Excel format. Now, on top of this, the other things that we're really working into here is we're giving the ability to expand upon what is included in our export beyond just the data. One of the questions that we get a lot of times is that, what does this field mean? How do I interpret this? What should be in these fields and whatnot? So we're including in our Excel exports explicitly this help sheet here, which describes each of the fields in depth. You can see, as you can see here, this is the first field in the page, which is the epitope of the IEDB IRI. It has the section, the header. We have 2 example exports, which should be continuous across the entire sheet here. And then we have another possible one, which just shows possible values that could be in those, because we don't have records that have every field filled out, because it just wouldn't have everything for every field for one particular record. Additionally, we also have this description field here, which is a short description of what that field means and how it's relevant to everything in the IEDB. And then also we have a link here for particular fields that have IEDB help links to describe it further or give a little bit more information about that field within the help documentation that's already available on the IEDB. So you can see this one goes to our IEDB curation wiki that shows what this epitope object actually means. So you can go there and get a little bit more information. One of the other things we are adding into this Excel export is the ability to capture your query parameters as well as the date that your file was generated, because these normally get lost in our current exports because we just have the raw data, and then it's up to the user to remember what that query that they ran encompassed there. So this is a really helpful little tab here for keeping track if you run multiple queries and run them, week to week or month to month to see the changes in the IEDB database for your particular information. So I've also got an example of the JSON format that I was showing. That this is more of a programmatic consumption for the data. You can see here that it includes also the export date and the search parameters, just like the Excel one, whereas the CSV and TSV formats do not have that information because they're just strictly data. But you can see that the formats here is the same the same fields as in those Excels, just formatted in a JSON format for consumption. Usually these are consumed by machine programs or websites if you want to feed them into something else. And I think I went really quickly through this.
Excellent. Thank you so much, Kelly. And apologies, I missed your introduction, but thank you very much for that. So that was the new database features, custom exports. We are running quite a few minutes ahead of schedule. So I'll pedal back a little bit and give you all some time to take a look at the questions that are coming through the Q&A function. So thank you all for that. But first you heard from Randi, who presented accessing the data, query reporting and examples. And those examples that Randi showed were actual user submitted requests as well. In addition to the screenshots that we showed, some of those queries were actually requests that were presented to us by our users. So that is just one example of how we do try and provide you, useful information in a research setting as well. Now, everything that Randi showed you is available currently online on the IEDB with all of those new features, with all of those existing features, I should say. The following presentation, which was new database features, custom exports by Kelly, our senior software engineer, that feature is still in development. So we are still testing this out, still refining, making a few changes to that work, but we're very excited to be able to show you a bit of a behind the scenes view on what that would look like as well. So basically you're getting a first glimpse at something that will be coming in the next few months to the IEDB. Once that is available to you all, we'll be sure to post that on our homepage, red banner at the top to let you know that it's live, but you all just got a behind the scenes glimpse for the first time. So with that now, we do have quite a few questions and we do have time as well. So feel free to take some time in responding, but I would like to invite Randi back to the virtual floor. We'll also have Sidne joining us, who is one of our scientific curators on the team that will be supporting Randi in answering those questions. And then Kelly as well to answer any custom exports related questions. So Randi, would you like to take that first?
Sure. So the first question I have is, do you have data on experimental mutations within epitopes and data on natural variations? This is from Chris Thorpe, and I'm going to share my screen. We have any data that gets published. So if they publish on a sequence that contains a mutation, whether it's natural or not, we will curate it. And so using the epitope search box, as I had shown in my query, you can use these blast options to broaden your query. So here I broadened it to blast 70%, and I put in a well-known epitope. And you can see all these different variations, and some of them are natural, and some of them are just longer. But there are some natural variations that you'll tell by there being an antigen. In the cases where there is no antigen, those are artificial. And then you also have the ability to search, as I showed in my talk. You can specifically request that you get data on analogs. And neoepitopes will be natural variations also. Okay, Sidne, I think you're up next.
Hi, so the next question is from Adrian Noah Bolano, and he was asking, could it be that you have the antigen registered there, but no data for assays, epitopes, etc.? And then you added on later about human and endogenous retroviruses. I don't know if you have a specific antigen in mind, but the IEDB and what we do is epitope driven. So unless there's an epitope, an antigen is not going to be curated because all the experiments are designed to describe what has been done with the epitope with respect to that antigen. I did do a quick search for human and endogenous retroviruses on our website, and we have 3 of the HERVs organisms curated. One in particular, HERV-H, I just looked at, and it has 1 epitope associated with that virus, it belongs to the envelope protein, and there's only one assay, and it happens to be a negative assay. So if you were only searching for positive assays, you wouldn't have seen any information.
Okay, you want me to go next? So next question I had. Can the positive T cell assay number versus all tested assay numbers also be displayed on the epitope query page? So also I wanted to quick query. So how that might be difficult for us to do. So when you see that assay details page like this that has these positive and all, this is a pre-computed page that gives you a summary of all the data in the IEDB. But when you're doing your query and you're looking at the data, you're seeing the data as presented based on your query results. And these counts over on this page are related to the query results based on your query. So here everything is positive because I only asked for positive assays. If you included negative, it could possibly tell you how many were positive and negative. But the important thing to note is there's so many different assays captured. This is an awful lot of information for us to try and make specific to your query and put onto that page. It would really clutter it up and complicate the view. And we're trying to keep the main result pages, too many windows open, sorry. Trying to keep the main pages as simple as possible. But if you export the results, you can always get those counts on the fly. Hopefully that helps. Okay, I think Sidney,
the next question we have is from Patrick Wu. Does the IEDB capture all the HLA typing information included in studies? For example, consider a study that reports ELISpot data using cells from subject A. The study also includes subject A's HLA typing information. HLA DRB1 allele information was reported as DRB10404 and DRB11301. So we try. For the most part, if they provide specific allele information for a particular epitope, then that would be included as the MHC restriction with the level of evidence being provided as well, whether it was just cited by the reference, whether they actually did binding studies to show that that epitope bound that particular allele and so on. In the cases of where it's more of a general thing, so you're giving us class 2 alleles, if there's only one subject being tested and the epitope was reported to be restricted to class 2, we can and try to enter the subject's HLA typing within the curation. So that information would be in the epitope readout or results page. You can imagine that that becomes untenable when there are multiple subjects carrying multiple alleles, and the epitope is still only restricted to class 2. In those situations, if we can find a common allele to all the subjects that respond to that particular epitope, we will try to put that in as an alleles present in the subjects, as opposed to an actual hardcore restriction for that epitope. I hope that makes sense. But we do try to put in as much information as we can.
And I'm going to answer, can't tell if it's an I or an L, Layden Makfar's question of what is the difference between B cell and T cell assay MHC ligand because the peptide that could bind MHC molecules could also activate B or T cell. So that's true. So all we have is a reflection of what was published. So if a peptide was tested in a B cell assay and a T cell assay, we would have both of those experiments in the IEDB. If an epitope was tested in 10 MHC ligand assays, 2 T cell assays, and 1 B cell assay, we would have all of those experiments in the IEDB. We just reflect what authors have tested. And if something's never been tested, we don't know whether it would be recognized or not. And many epitopes are context dependent. Like I said, they may be recognized in a diabetic subject, but not a healthy one. They may be recognized as a B cell epitope in bulb C mice, but not C57 black mice. So all of the experiments that we have in IEDB just reflect what authors published. So we don't know whether or not they could also activate B or T cells if they weren't tested for it. Do you want to go, Sidney, or are you done?
I think I am done.
Okay, then I'm going to finish up. I have one really quick one, which Alejandro Pina asked if I could elaborate more on the results of the Immunome browser. And I'm going to leave that to Alba because she's going to go into much more detail than my talk went into. I just wanted to point it out to you that it existed and it's a cool feature, but Alba will explain all of the details. Bjorn.
So there was one question from Mario Ostrovsky. Do you have HLA subtype frequencies listed in various populations in this database, or is there one you would suggest to use? So we have the population coverage tool, which is using HLA frequencies that we are extracting from a website called allelefrequencies.net. And we've been using that for over a decade. And we are actually right now in the process of switching. So the first answer would be allelefrequencies.net. And I think going forward, we are actually in the process of updating our source for this. And hopefully within the next year or two, we will actually have these things live, because like you, we've been having trouble finding this, and we have not been always particularly happy because that website is more focused on like, how do you say, population migration and those kinds of things of MHC frequencies in like in whatever mountain tribe populations or so. But yeah, it's actually very hard to find like very common population frequencies, and we are now switching to databases that are maintained by the transplant providers that have by far the largest and most detailed HLA typing information in different worldwide populations, which for our typical use cases is better. So, and these data are somewhat hidden, so we're going to try to make them more public, but there's papers if you're interested, like I can look them up, just email at help. But it's also very easy to find it on PubMed. It's just kind of hard to access the data. Sorry, that was a little long-winded, but I hope it's helpful.
Okay, and I'm going to take the one last question, which I don't have an answer to, but it's still a cool question. Mauricio Reggio asks, would it be interesting to have a column with an immunogenicity gradient score based on the number of positive and negative assays for a specific epitope in respect to the total of assays? So the concepts of immunogenicity and antigenicity scoring is really cool, and it is something we talk about. It's hard, again, because everyone's query, you come to the IEDB and you're interested in something different. So the response would have to be changed based on what the query results were. So if you're only interested in sheep, you're going to get a different result than if you're only interested in human data. But it is something that is interesting and that we keep on the conversation topic list for things to do and things we think about. So it will be noted down, and we will keep that information up as something we are definitely thinking about. It's a tools question. I think we can save that for later.
Actually, I don't think this is a tools question. Just because we have time, I'll answer it now. Daniel Hakim asks, when I search for MHC2 restricted epitopes, it lets me search for epitopes of length 9 or so. Net MHC2 pan uses a length of 15, as far as I can tell. Do flanking peptides matter for MHC2? So the first thing I have to point out is when you search in the IEDB, the length field is completely decoupled from the MHC restriction field. So you could search for something like MHC2 restricted epitopes and length 3, probably, and you shouldn't find anything. But there's no link in the search that links length, and you just not find any results. Actually, I do also have to point out that we have partial epitopes, which is a rare thing, and for T cell really shouldn't happen, but for antibody epitopes, it's quite often that you might even have just a single residue that is known to be part of an epitope. and then that's curated as like a partial thing. This could be like if you know escape mutation abrogates binding of an antibody, it would be captured as such. Now, the reason for net MHC2 pan using 15, or in general, like us liking to look at longer peptides for class 2, typically a peptide that is less than definitely like 11 or lower will not bind MHC class 2, period. The core binding region is around 9 residues, but there is a requirement for some flanking residues for backbone interactions, and otherwise things will simply not bind. Even though the core 9 residues are what makes direct contact with the binding groove, you do need longer peptides, and 15 tends to be the optimal length that is most frequently found.
Thank you very much for that, Bjorn. We do have a couple of last minute questions come through. I will give the team just a minute to take a look.
Okay, I'll take one. Tian, do you think 2 different viruses can have the same epitopes of human? For example, influenza and EBV have the same epitopes of HLA-A2. Yes, we say that all the time, that there's the same sequence conserved across viruses, but also the same sequences recognized in both mammals and in viruses in autoimmune situations. And if you did a binding query on any MHC, you would get all of the epitopes shown to bind that MHC in any of the publications we've curated, and you'll find all sorts of different sources, and you can analyze them to see how many of them are identical or similar.
So I thought I'd do the last one here, which is the shortest epitope to be positive outcome used for diagnostics. So I'm answering this because it kind of relates to the one I just had before. So in a diagnostic setting, you would like have peptides and look for them being bound by antibodies or triggering a T-cell response. So what we would normally consider is if these are linear peptides, something like 10 residues for antibodies, something like 8 residues as a very minimum for class 1, something like 13 for class 2. That's been what we've been using in the past, and typically a little longer is always better. And with that, I think we should stop it here.